B cells. So the way B cells get activated is that they bind to a specific antigen. So say there was a green one here, this one would not, this is a receptor. This receptor will not take it because it doesn't bind to that antigen. So
it has to match.
It has to match. It has to be specific. Once a cell is activated, it starts cloning, and it also makes a memory. These clones are gonna differentiate into plasma cells. And then these plasma cells are the ones that are gonna make the antibodies that are gonna help attack. But they do not kill. No B cells ever
It doesn't kill, it just
They
attacks.
don't kill.
Okay.
This memory cell is gonna help you. Say you see that antigen a second time, which is gonna help cloning go a lot faster. It's also gonna make the cells live a lot longer and attack more forcefully. Okay, and then we can go.
That's the second time around because it's
The second time around, it's more forceful. And then we could see the helper T cells. Helper T cells come from CD4s. This is a APC, which is an antigen presenting cell. There are three antigen presenting cells, which is a macrophage, dendritic cells, and an activated B cell. Notice I said activated, so it has to be activated in order to work as a APC. So this is an APC. This one is gonna take an antigen from the outside. So that's why it's called exogenous.
Just indented and activated.
Yes. It's exogenous because it's getting the antigen, it's coming in from the outside of the cell. So it's gonna take it in, and then it's gonna lysome it, which is gonna break it apart. And then it's gonna, the APC is gonna put a fragment of the antigen in the MC2.
Oh, it's gonna bind that to the MC2.
It's gonna put it down here.
Oh, bring it out.
Yes. It's gonna put a fragment of it. Not the whole thing, just a fragment. Yes. And
so these are cloning in memory. So it
Yeah.
does the same thing but faster.
it's activated already, so because it's activated, it's gonna clone again, and it's gonna make memory.
Okay.
Yes. So another thing to point out is that helper T cells only bind to MCH2. They do not bind to MCH1. Only cytotoxic T cells bind to those. So a helper T cell is gonna bind to the MHC2. It's gonna bind there. The way that it is stick together, that way it doesn't break apart, is by the CD4, which is a protein. So it makes sure that they stay connected. So once it's activated, like I said, it's gonna clone, it's gonna make memory, but this one is gonna release cytotoxins.
Okay, the cloning cells in the cloning cell release
Cytokines. There's two different types of cytokines. Yes. And these cytokines are chemical messages and they're instructions for other cells.
Other immune cells.
Other immune cells, yes. So these cytotoxins are going to increase permeability, vasodilation, phagocyte mobility, and the proliferation, which is the rate that they reproduce. Okay? So they're gonna co-stimulate here. So that's what helps it clone faster. And it's also gonna co-stimulate here. Now this is a T cell, a cytotoxic cell. So these come from the CD8 proteins. So this is another APC, which again is a macrophage, a dendritic cell, or activated B cell. It has to be one of those. So if you guys notice here, there's a MHC2, which binds to the helper T cell, and it's gonna connect by the CD4. So it's gonna activate. So once again, it's activated. It's gonna clone. It's gonna make memory, and it's gonna make cytokines. And then so this helper T is gonna signal the APC to put a fragment on an MCH1. What binds to the MHC1? The cytotoxic.
So MHC1 only binds with cytotoxin.
That's
and
correct.
MHC2 only binds with helper T cells.
That's correct. And then what it holds them together is the CD8. It's different for this one and this one.
Say that again.
It's different for the
4.
CD4 and CD8. CD8 is only for the cytotoxic. So once again, it's activated. It's gonna clone. It's gonna make memory, and it's gonna have cytokines. But the difference between this one, it's not going to release them. This one releases them.
Okay, the T helper cells release.
Yes, you saw how it released it here, and it co-stimulated.
Okay.
This one's not gonna release it yet.
Okay.
So it's gonna make an army, and then it's gonna go out to the body, and it's gonna look for infected cells. So once it finds an infected cell, it's gonna bind to it, and it's gonna release its cytotoxins. And there's 2 chemicals that are being released, which is no, this is different. There's perforin, which makes holes that allows the granulocytes to come in.
So it will make perforin, and then it makes this one too. 2 of them.
There's 2 different that are released. This one's gonna make a hole in the cell, which is gonna allow the entry of these enzymes to come in. And another thing I should point out, like I said, over here where we put the fragment of it, this one is gonna make a protein, so it's gonna be infected from the inside. So it's gonna make a protein that's abnormal, which is also foreign, and it's gonna stick out the whole protein.
So on the healthy one? On the infected?
On the infected one,
Okay.
yes.
You'll only look for the unhealthy one, so the infected one.
Yes, and another thing I should point out, the MHC is looking at that self, that's me. The protein is looking at, which is this, am I infected or am I healthy? And that's about it.
Anything else before I stop recording?
That's it.